The title of our video is The Gels Entity, which refers to a group of entities that act like a single entity, and the subtitle of our video is How Consciousness Emerges in the Human Brain. We use the word like with caution, meaning it appears to be so, but not actually so. Each unicellular organism possesses its own unique life and realm of perceptual awareness, and different types of unicellular organisms likely have different realms of perceptual awareness or consciousness, based on their capabilities, situation, and location. Similarly, each cell of a multicellular organism possesses its own distinct life and realm of perceptual awareness or consciousness, depending on its position, situation, capabilities, and location. The core theme of this theory is very clear: I am a single neuron cell located in the pulvinar. There are some special kinds of functional neurons in the pulvinal, I am one of them. Since I am a single neuron located within the pulvinar, there are, however, several other identical neurons right beside me, within the pulvinar, that have the same perception or awareness. That means, they are thinking and feeling almost the same things, in the same way, at the same time as I am. The mystery here is that we don't comprehend the issue, or it could be said we're not accustomed to thinking in this way. For convenience and clarity of exposition, the research process has been organized into two major sections. Here, Section A presents the main exposition, while Section B addresses general queries. Section A. The key analytical insights are outlined below. 1. Each cell of a multicellular organism possesses its own distinct life and realm of perceptual awareness or consciousness depending on its position, location, capabilities, situation, and nature of information. These criteria hold true for neurons as well. 2. As per the outcome of number 1, with reference to Part 1 and Part 2, since I am a single neuron cell with visual, auditory, tactile, gustatory, olfactory, nociceptive, and hedonic perceptions, it should therefore be located in a brain region where all related information is accumulated or converges. I am one of them in that cluster of specific functional neurons. At that location, what is my specific function as a single neuron? In answering the question, the main clue comes from visual perception and the flow of its associated information pathway. Section B. Some general queries that might be on our mind are as follows. 1. Except for my identical cluster of neurons in similar locations with similar functions, what would be the perceptual realms of neurons in other brain regions? 2. Is it, my entity/neuron, replaceable? 3. Finally, can this theory be used to argue against or in favor of the belief in the existence of God? Description of Section A. 1. The blood cells in our body have their own distinct life and a lifespan that is separate from the human lifespan. If these blood cells are transferred from one person's body to another, they can continue to live and function in the same way as they did before. Similarly, the different parts of our body can be transferred from one body to another, and they can survive and function in the same way in another body as they did before. Since a cell can be transferred from one person's body to another's and can continue to survive and function in the same way as before. Therefore, each cell of our body must possess its own distinct entity, life, and perceptual awareness, at the basic level, until it is considered a dead cell. In multicellular organisms, cells are organized into different groups, which then divide into various subgroups and further into sub-subgroups, functioning in a hierarchical and specialized manner. That means, different clusters of cells, including different clusters of neuron cells, have their own distinct realms of perceptual awareness or consciousness. The extent to which this basic level of perceptual awareness in any individual cell of a multicellular organism will ultimately reach a level of self-perception, self-awareness or consciousness depends on its position, location, the type and amount of information it receives, and its ability to exchange information. In case of a neuron cell, the basic level of perceptual awareness of an individual neuron, eventually, at some stage, emerges to a level of self-perception, self-awareness, consciousness or full consciousness depends on its position, location, the type and amount of information it receives, and its ability to exchange information, where each group, same brain region with same functional cluster of neurons, possesses its own realm of perceptual awareness, consciousness or full consciousness. The meaning of full consciousness is relative to that of a fully conscious human being. We often completely forget that multicellular organisms originated from unicellular organisms, or developed from a unicellular phase, where each cell of a multicellular organism possesses its own distinct life. When multiple unicellular organisms unite to form a multicellular organism, they gain various benefits, and their chances of survival increase greatly. At this stage, the cells in a multicellular organism become functionally specialized, resulting in a significant reduction in both their individual functions and the functional load that each cell carries. In Part 1 and Part 2, it has already been explained that each cell of a multicellular organism possesses its own distinct life and realm of perceptual awareness or consciousness, based on its position, location, capabilities, and nature of information. These criteria hold true for neurons as well, and I am a single neuron cell located in the pulvinar. In the case of consciousness emerging in a human brain, nothing is being imposed or created separately in our brain, and nothing supernatural is happening there. Description of Section A, Number 2. Since I am a single neuron cell with visual, auditory, tactile, touch, gustatory, taste, olfactory, smell, nociceptive, pain, and hedonic, pleasurable or emotional, perceptions, it should therefore be located in a brain region where all related information is accumulated or converges. I am one of them in that cluster of specific functional neurons. Figure 1. The green and cyan blocks in this figure denote the multisensory perceptual areas of the human brain. Here, all blocks play an important role in visual processing. The left and right sides are denoted by L and R, respectively. Figure 2. This figure illustrates the connectivity of multisensory receiving areas, including the pulvinar, showing both their internal connections and their pathways to other key visual processing areas. Blocks are represented by their relative position in a two-dimensional image. Sparse connectivity is not depicted in this figure. Among these multisensory regions, where is the specific neuron, my entity neuron, located? Here, main clue comes from visual perception and the flow of its associated information pathway. The human retina transmits visual information, or receptor data, to the brain at a rate of about 24 million visual signals per second. Considering that an average retinal ganglion cell fires about 10 spikes per second, it can practically range from 5 to 40 spikes per second depending on the cell type and the stimulus. If depth, for a three-dimensional view, pixels address, or object address, and orientation-related data are included, the visual system has to process several million visual signals per second, amounting on average to at least 26 million neural spikes per second. If auditory, tactile, gustatory, olfactory, nociceptive, hedonic, and the meaning of their information data are included, it requires an additional receiving capability of at least 3 to 5,000 spikes per second for a single neuron to perceive. However, this additional amount of data is actually negligible in comparison with visual information. It can be noted that the meaning of data arises only after comparison with retrieved memory traces, a process that demands a huge amount of neural computation. Such data comparison involves a distinct processing pathway. On average, a neuron in the human brain can have 10,000 to 40,000 synaptic connections serving as inputs. A large number of synaptic connections serve as outputs, which are not included here. If we consider a normal neuron firing rate of 10 per second, then the total maximum number of spikes a neuron can perceive is about 0.4 million per second. This is a max value under normal conditions, but practically this value will be much lower. This difference between 0.4 million and 26 million remains very large, even under a conservative approach. This implies that, without different technical aids, it is impossible for a single neuron in the human brain to directly utilize this vast amount of visual information and then perceive the three-dimensional outside world. That is why the main clue comes from visual perception and the flow of its associated information pathway, which can help identify the location of the specific neuron, as well as the cluster of neurons. With the help of various technical aids, the required amount of information per second can be brought within the limit that a single neuron cell can perceive the outside visual world. First of all, practically, we perceive fine details of only about 1% to 2% of our total visual field. Saccadic eye movement helps to overcome this visual challenge. Secondly, our brain filters important elements from various others by concentrating on them. Thirdly, different areas of our brain are specialized for different tasks, like object recognition, motion detection, and assisting in decision-making, etc. After this processing, the result goes to the relevant destinations. Fourthly, during the processing of information, some additional information is added before and after each spike, which helps perceive the clarity of the information by characterizing it. Altogether, these can drastically reduce the required amount of information per second that can be perceived, all information including outside visual world, by a single neuron, as well as by the neurons of a specific cluster. Figure 3. This figure illustrates the retinogeniculosstriate pathway, primary visual pathway, from the retina to the primary visual cortex via the lateral geniculate nucleus, LGN. Blocks representing three-dimensional brain regions are shown here according to their relative positions in a two-dimensional image. Figure 4. This figure shows the connectivity between corresponding functional areas in the brain's left, L, and right, R, hemispheres. This diagram illustrates the connectivity of visual processing and multisensory integration areas, excluding the pulvinar. Sparse connectivity is not depicted in this diagram. This diagram illustrates the connectivity of multisensory and visual processing areas, including the pulvinar. Sparse connectivity is not depicted in this diagram. This diagram synthesizes observations from thousands of experts and clinician comments on patients with strokes or other injuries, focusing on their functional characteristics and anatomical connectivity. We incorporated only the data that is relevant to and supportive of this theory. It is important to note that, due to its unique connectivity and anatomical position, the pulvinar is the best-equipped region for all 7 modalities, which it can receive, perceive, and use to set prioritization, a process involving steps for decision-making. The current understanding of the principal multisensory and visual processing areas is described below. You may pause the screen to read the brief descriptions. The multisensory and visual processing areas include the midbrain, mesencephalon, superior temporal sulcus, STS, inferior temporal cortex, IT cortex, posterior parietal cortex, PPC, prefrontal cortex, PFC, anterior insular cortex, AIC, and amygdala, while the visual processing areas include V1, V2, V3, V4, and middle temporal visual area, V5 alias MT. The fundamental biological neural network and the associated mathematical relation are shown here. They are applicable to all regions of the brain, where the input neurons of one region can also act as the postsynaptic neurons for other functional regions. A person can still have complete visual perception even in the absence of some of these regions, for example, when one is damaged. However, this often results in an inability to process certain other application functions. Another important issue is that these regions are functionally specialized and highly divided, and many do not receive the necessary retinotopic mapping or visual signals. No single neuron within a cluster of neurons in these areas has been found to exhibit multisensory perception of all 7 types. Consequently, none of the areas meet the criteria for the target region. Pulvinar. There are many other brain areas involved in visual processing, each with a specialized role, but the pulvinar is unique in certain ways. No other area does exactly what the pulvinar does. Its role is more about orchestration and control rather than raw feature extraction. The main function of the pulvinar is to set prioritization in the decision-making process. The pulvinar is the only area in the human brain that has strong direct connectivity with all types of visual information processing areas, including retinotopic mapping signals. The human brain is confronted with the computational challenge of interpreting millions, about 26 millions per second, of concurrent visual signals to construct a coherent perception of the external world, which is a formidable task when considered against the relatively limited functional capabilities, with relatively constrained informational bandwidth, of an individual neuron. Auditory information is a time-based signal that ranges within a few kilohertz. The rest of the tactile, gustatory, olfactory, nociceptive and hedonic signals altogether can be covered within 100 Hz of neural spiking. In a manner similar to visual information, the pulvinar also receives processed auditory, tactile, hedonic, olfactory, gustatory and nociceptive information, whether integrated or non-integrated, from different areas of the brain, such as the MGN, medial geniculate nucleus of thalamus, VPN, ventral posterior nucleus of thalamus, PPC, PFC, OFC, STS, midbrain, amygdala, IT cortex, and AIC. For instance, IT enables the grouping of objects into categories, such as animal, tool, vehicle. So, if IT answers what it is, then STS answers what it is doing in a social or biological sense, and MT answers how it is moving. While PPC replies where, PFC answers what it means, and AIC adds how it feels right now. When a person perceives that a ball is above the head, relative to the eyes, head, hands, and body, and then as the hand approaches the object, the PPC helps determine body position and guide the hand for accurate movement. Almost all sensory information passes through the thalamus before reaching the appropriate cortical areas, except for olfactory signals. All ascending and descending pathways, as well as the reticular activating system, RAS, pass through the thalamus, except for the autonomic system. The pulvinar, a part of the thalamus, is exclusively connected with some other thalamic regions, playing a central role in filtering, integrating, and determining priority for other brain regions. In this prioritization process, the pulvinar also directly communicates bidirectionally with the PPC, PFC, OFC, STS, midbrain, amygdala, IT, AIC, and other regions. For example, if a person is searching for her blue bag in a crowd, the pulvinar first boosts blue-colored features through prioritization, the PPC shifts attention across locations, and once the bag is found, the PFC maintains the goal, blue bag, by suppressing other elements. Each neuron within any specific functional group in the PFC does not see the image. Like an AI neural network, the PFC extracts specific features and elements from the image by utilizing memory. Then, the pulvinar brings the blue bag from the mind into real visual perception. Here, with the help of other regions of the brain, each neuron within a specific functional group in the pulvinar can see the image, facilitated by a reduction in the number of spikes per second. In that case, the 26 million, gross, units of visual information, retina to LGN to visual cortex, per second are reduced to within thousands, less than 400,000, per second by using preprocessed information. Concentration, for extracting only relevant elements, eye movement, to focus on fine details occupy only 2% of the total visual field, and by adding signals before and after each spike for clarity of information. The process here emphasizes the perception of objects in the peripheral zone over the perception of pixels. The number of visual information signals is reduced by the PPC, PFC, OFC, STS, midbrain, amygdala, IT, AIC, V1, V2, V3, V4, MT, etc., which are exclusively engaged in visual processing and information integration, and these regions help the pulvinar with preprocessed information. The pulvinar also mutually helps them by synchronizing neural activity between perceptual and executive systems. The pulvinar also influences the RAS, reticular activating system, when needed. With extensive connectivity, the pulvinar can control which information is prioritized in the decision-making process. For example, tasks like typing, cycling, walking, driving, eye movements, and language fluency can be performed under the autonomic or semi-automatic, subconscious, system once the brain has trained itself to run them under this autonomic or subconscious system, by utilizing other brain regions. However, the pulvinar can pull these tasks back into full attention when required. Then, the autonomic and semi-automatic, subconscious, systems become part of the prioritization process by suppressing or withholding activation from areas that are not relevant to their functions. It is essential for the effective functioning of an individual. Otherwise, any large and complicated system would not be able to function and would become ineffective or useless. To use an analogy, in a large construction project, a director cannot be involved in every decision or task. The director must engage hundreds of experts, such as engineers, CAD designers, and skilled workers. One person cannot do others' jobs, category, perfectly. The director must rely on approved experts, giving them the freedom to work according to their judgment under the director's main guidelines. In this analogy, all working processes become part of the main executive system, otherwise, a large system cannot function at all. In a similar manner, the autonomic and semi-automatic systems of the brain are part of the prioritization process. In fact, this functioning process is essential for an individual's survival. The terms prioritization and decision-making are closely related but refer to distinct cognitive processes with different purposes and mechanisms. Prioritization is the process of ranking tasks, options, or needs in order of importance, urgency, or value. It is often used when multiple tasks must be done, but not all at once. Decision-making, on the other hand, is the process of choosing from one or more options to achieve a goal or solve a problem. Prioritization can be a step within the decision-making process, providing a guideline for the final decision. Under certain conditions, especially during important decisions, for example, avoiding threats, or when something seems significant. The pulviner's rapid prioritization may almost act like decision-making by quickly biasing the system toward a specific perceptual or behavioral outcome. In this situation, the pulviner directly exerts control over the decision-making process. The pulviner occupies a unique central anatomical position in the human brain. Structurally, it sits in the central posterior part of the thalamus, and functionally, it serves as a central communicator and integrator for distributed brain systems. Owing to its direct connectivity with all major visual processing areas, multisensory integration regions, involving two or more modalities, and other thalamic nuclei, the pulviner can synchronize neural activity between perceptual and executive systems. It can be noted that multisensory limbic integrated information can be incorporated into the resultant outcome from 7 fundamental streams: visual, auditory, tactile, gustatory, olfactory, nociceptive, and hedonic. The unique anatomical position and connectivity enable the pulviner to serve this role. For instance, a person feeling hungry and going to eat may have their decision-making process shifted by bad news due to a change in prioritization. The person may not feel any hunger in the new situation. Then, through prioritization, the pulviner can regulate brain activity by switching between competing stimuli during the decision-making process. Thus, the pulviner plays a unique role in the orchestration and control of different brain regions, rather than in raw visual feature extraction. Due to its unique connectivity, anatomical position, and bidirectional communication, the pulviner is an exceptionally well-equipped region to serve as the key multisensory hub, where information has already been integrated with different combinations of modalities, and features have already been extracted before reaching the pulviner, of all 7 modalities, including multisensory limbic integration, for perceives information and setting prioritization. Therefore, the pulviner does meet the criteria for the target region, and it has a convergence zone where single neurons can receive, see the complete image, and perceive pre-processed information from all 7 modalities, which is eventually implicated in setting prioritization. Description of Section B. Some general queries that might be on our mind. 1. Except for mine, a neuron, an identical functional cluster of neurons, what are the perceptual realms of neurons in other brain regions? Each cell and each similar functional cluster of cells in our body has its own separate perceptual realm. For instance, blood cells, skin cells, and other cells in our body have their own distinct lifespans and perceptual worlds, depending on their location, situation, and other factors. Similarly, each neuron, that is, each neuron of a specific functional cluster, has its own distinct perceptual world, depending on its location, task, situation, nature of information, and other factors. That means each neuron has its own distinct realm of perceptual awareness, consciousness, or full consciousness. Here, the meaning of full consciousness is relative to the fully conscious activity of a human being. This concept can be illustrated through the analogy of a television screen displaying a movie. The processes occurring behind the screen, that is, the signals within the television circuitry, are fundamentally different from the visual output presented on the screen. Within the circuitry, distinct functions are distributed across specialized components, and each function operates independently without being directly interchangeable with others. However, when integrated, these separate functions collectively generate a coherent and meaningful output. Similarly, neurons across different brain regions maintain distinct perceptual realms determined by their location, functional role, situational context, and the nature of the information, etc. Due to the lack of large-scale multidisciplinary research, no detailed empirical data are available. However, many scientists believe that it is entirely possible for each neuron of a specific functional cluster to receive indirect, pre-processed information from all 7 modalities. Section B number 2. Is it, my entity/neuron, replaceable? Although the replacement of a neuron is theoretically conceivable, in practice such a procedure is currently unfeasible and is unlikely to be achievable in the near future. This limitation arises primarily from 2 factors. First, each neuron forms thousands of highly special microscopic synaptic connections, the precise reconstruction of which is extraordinarily complex and practically impossible. Second, it is not possible to externally verify whether the replaced neuron truly a new entity. This is because, who will tell others, investigating persons, that it has been replaced? Actually, the replaced neuron becomes a replica and integrates into the existing population of thousands of identical functional neurons. It is a highly profound and insightful query. Section B number 3. Finally, can this theory be used to argue against or in favor of the belief in the existence of God? This theory is not capable of providing any idea about the existence of God. However, it indirectly suggests that the entity does not cease to exist after death. In other words, it implies that the existence of the entity persists at a more fundamental and elementary level. It brings to mind the statement often attributed to Albert Einstein, "I believe in a God who manifests himself through the harmony and order of nature." Conclusion. From our current understanding, there is no single neuron, that is, no neuron of a specific functional cluster, in the human brain, except the pulvinar, where the required visual information, including the requirement of minimum retinotopic mapping signals, can be received and perceived. Again, based on our current understanding, there is no single neuron, that is, no neuron of a specific functional cluster, in the human brain, except the pulvinar, where all 7 types of information or integrated modalities, visual, auditory, tactile, gustatory, olfactory, nociceptive, and hedonic, including multisensory limbic integrated information, are accumulated or converged. Due to its unique connectivity and anatomical position, the pulvinar is the best equipped region to serve as the key multisensory hub of all 7 modalities, with multisensory limbic integrated information and required retinotopic mapping signals. Thus, specific functional neurons of the pulvinar can see the complete image, perceive all this related information, and set prioritization. Therefore, this theory concludes, I am a single neuron located in the pulvinar. Neurons of the human brain are organized into different groups, which are further divided into various subgroups and sub-subgroups, denoted here by Zell's entity. Each neuron of the human brain possesses a basic level of perceptual awareness, which eventually, at some stage, reaches a level of self-perception, self-awareness, consciousness, or full consciousness. Depending on its location, position, the type and amount of information it receives, and its ability to exchange information. In the case of consciousness, nothing separate is being imposed on the brain, nothing is created separately, or nothing supernatural is happening in the brain. Due to the limited scope of this research, broader multidisciplinary investigations could not be conducted, therefore, some specific empirical data are lacking. To obtain such data, large-scale multidisciplinary research is required, involving leading experts from diverse relevant fields. Clearly, this theory is set to open a new window, enabling us to find answers to many difficult unanswered questions. Thank you for watching.